by talking about calico cats and how this is a form, and we also talked about twins briefly, differences between identical twins. We've got calico cats, which is apparently the only reason that some people showed up today, but that's fine. Cat's part of the lecture, so that's fine. Just kidding. There are no lolcats in here. Sorry. Start off with a brief video. This is going to tell you most of what you need to know about the concept, then we'll get into the details. Brief video, like a minute.
And these cells start off in a female with 2 X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still, each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one the maternal one. And now, this process will happen at random, and you'll have almost half of the cell with the maternal X active, half of the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this in mosaic appearance, because of the process of X chromosome inactivation.
So that's X chromosome inactivation, which is abbreviated XCI in the movie. So X chromosome inactivation. This is something that happens in which sex in humans? Both sexes or just one? Do males do X chromosome inactivation? Right. Males, how many X chromosomes do we have, guys? Just one. Each of our cells has one X chromosome. So we've got XY males, and we've got XX females. So as this video suggested, what happens during early embryogenesis, in the first few cell divisions of life, some point early on, in females, one of the 2 X chromosomes gets turned off forever. That's what's termed X chromosome inactivation. That's what they were trying to demonstrate with one X chromosome starting looking like a full-size chromosome and then winding up being a little straight line. So I'll play this video again after we're done talking about details of X chromosome inactivation. Why is X chromosome inactivation critical? Why does it happen? Why do females turn off one of their X chromosomes and not males? So,
No, go ahead.
both.
No, go ahead.
Should we flip over it? So what did you say?
Because males only have one.
Okay, so if males only have one X chromosome, what happens if we undergo random X chromosome inactivation? Which X chromosome do we turn off? The only one we've got. And if we inactivate our X chromosome, that would be, unfortunately for us, really bad because the X chromosome has genes we need to survive. So males don't do X chromosome inactivation. But maybe we should back up a little bit. What's the purpose of X chromosome inactivation then?
You don't double the expression.
What would happen if females didn't randomly inactivate one of their 2 X chromosomes in every cell? Why is it important that men have 1 X chromosome and females have 2? What is that? It's not fair, guys. Literally, guys. They get 2 copies of an X chromosome. We've only got 1. So women have twice their diploid for the X chromosome. Males are haploid. We've only got 1 copy of the X. So it's imbalanced. Men have 1 X chromosome, females have 2. It's detrimental then to have, we've evolved to the point now where it's detrimental to have 2 X chromosomes being active at the same time. So through the process of X chromosome inactivation, one of those 2 X chromosomes at random, you never know if it's the one on the left or the one on the right, early in development gets turned off by being condensed and then condensed and then condensed into what we call a what? What does it see when you look at a nucleus and there's a cytologically, microscopically distinct little blob that is the turned off X chromosome? What's that called? It's not a polar, it's a something body. Named after that famous comedian from the 80s, Roseanne Barr. No, not Roseanne Barr. It's a Barr body. With 2 R's, like Roseanne Barr. Google it. You guys are too young. Named after a geneticist or microscopist that discovered this. So in every female cell that you look at, you can detect which of the 2 X chromosomes is inactivated because it looks like I drew it here. So at metaphase, you could see one X-shaped pair of sister chromatids, then you get a little blob, the Barr body. Highly condensed, really packaged with lots of proteins. That's the inactive X chromosome. That's how the cell turns off expression of genes on the X chromosome. Protein's basically wadded up into a little ball. We've got all day today to talk about how this happens and the molecular details. If anybody has any questions right now, it's a good place to start. So we'll wander through the slides over the next 40 or so minutes. But
So do they only have from one X chromosome, or do they have like a mix of them?
by mix, you mean, so in each cell, one or the other X chromosome is turned off.
Okay.
So when you say mix, you're
Like
talking
in the
about...
whole body.
Okay, so let's do this again. You're absolutely right, and that's the point that's made by this video. So let's watch again. So these are the 2 X chromosomes in a single cell.
The cell divides, and the embryo is getting
So this is
slightly
early
bigger, and
embryogenesis,
still,
no X chromosome
each cell
inactivation
has
yet.
both X chromosomes active.
And this is saying at
But
this
in early
stage,
embryogenesis, each
here's
cell
where one gets
will
turned off.
inactivate one of its X's, and one cell will remain with the paternal X active, while the other one, the maternal one. And now, this process will happen at random, and you'll have almost half of the cells with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat-color gene is on the X chromosome, and that's precisely why these cats look like this in mosaic appearance, because of the process of X chromosome inactivation.
Okay, so in other words, let's say it was the 2-cell stage where one of the X chromosomes get turned off. We've got X chromosome 1, and we've got the second X chromosome, which I'm going to show as a Barr body, just a little blob of DNA and proteins. And let's say in this other cell, this is way earlier than X chromosome inactivation happens, not at the 2-cell stage. Say it's the first X chromosome that gets turned off, and the second X chromosome remains active. When these 2 cells undergo cell division, what would you predict is going to happen? So when you look at the cell on the left, the second X chromosome is inactive. Is that pattern going to be true in both of the daughter cells? That is, do both of these 2 cells that result from division of that cell, do they both have their second X chromosome turned off, or is it something else? This is why it's called epigenetics. There's inheritance here, and in this case, which X chromosome is turned off is inherited from generation of cell to the next generation of cell. That is, after cell division, it's the same X chromosome that's still inactive after the first division. And the same is true over here. So the X chromosome version 2 was active, X1 was inactivated, and that pattern is passed on from cell to cell, from division to division, throughout the rest of embryogenesis and development. So which X chromosome, bottom line, take-home point, write it down, which X chromosome gets randomly turned off is heritable within that organism. Other questions at this point? So cell division, cell division, cell division. Once an X chromosome gets turned off, it stays turned off in all of the daughter cells that are produced. So then you get an adult that's got billions of trillions of cells. If I was a female, some of my cells would all be originated from one of these cells that initially turned off its X chromosome. Pretty straightforward. In what sense is this not heritable? I want to make this distinction clear because this can get a little bit confusing. What are we normally talking about? What process are we talking about when we talk about inheritance? When I say I've got a heritable genetic disorder, what does that mean to you? What does it mean when I say it's heritable?
You're talking about you're passing it on to your offspring.
So I could pass it on to my offspring. X chromosome inactivation is not that type of inheritance. That's not what we're talking about here. We're not talking about, does a parent who has their X chromosome 1, the mother, has her X chromosome version 1 turned off, doesn't pass that to her kids. That's not that type of inheritance. The inheritance we're talking about is just in cell divisions in the tissues of the individual. It's heritable from one cell to the daughter cells during cell division, but not from person to person, not from generation to generation. Subtle point, but important. Will come into play later. The goals for today, okay, we want to know how epigenetics is different from genetics. We talk about it in a genetics class because it has to do with the main purpose, what we're here to talk about today is gene expression regulation, continuing on from last week. How is epigenetics different from genetics? And then you should be able to predict, based on what we learned today about epigenetic marks, which are chemical modifications to DNA, whether or not you expect a gene would be turned on or turned off. So simple outcome there. So genetics, as I mentioned last class, is the study of DNA, especially inheritance from generation to generation. Right? Study of the sequence of DNA, study of the transmission of DNA, meiosis, mitosis. But it's about, and most of our focus this term has been on the nucleotide sequence, A, T, G, C. Right? The nucleotide code, in other words. But then there's something that happens to DNA on top of, or epi, the DNA. Those are chemical modifications. So we have enzymes in all of our cells that go around and take the A's, T's, G's, and C's and add additional chemical groups to them. You ready for some organic chemistry? Oh, hell yeah. No. Nobody's ever ready for organic chemistry. So we're not going to get too much into the organic chemistry. I just want to remind you about functional groups and methyls and acetyls and so forth. So these small little molecules, small little organic molecules, additions to the DNA, do something to gene expression. That's epigenetics. It's like enzymes, proteins, taking spray cans around our genome and spray painting, tagging. It's literally called tagging. Sticking on these little chemical modifications to our DNA, and that affects gene expression. So genetics has to do with the sequence of the DNA. Epigenetics has to do with the chemical changes that get made on top of whether or not it's an A, a T, a G, or a C. So I want to start by reminding you about chromatin structure. We've talked about this before. DNA is accessible up here in between where it's wrapped around these proteins called histones. Does anybody remember how many histone proteins make up a nucleosome? 8. Yeah. So there are 8 histones. For the win, what are the names of the histones? So there's histone, strangely enough, there's histones H2A and H2B, so the 2 flavors of histone H2. There's a histone called H3 and a histone called H4. That's only 4. So you multiply that by 2, and you get the 8 nucleosomes, or the 8 histones that make up a nucleosome. So there's 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s that all come together in a big blob, essentially. That's what's diagrammed here. So it's 8 histone proteins make up one nucleosome, and then DNA wraps around it, and we know that that's how DNA is packaged into the things we call chromosomes and chromatids. What does chromatin structure have to do with gene expression? It's a good place to start because we've talked about it before, and that's exactly how epigenetics works. How does DNA compaction affect DNA expression, whether a gene gets transcribed? What are the 2 different types of chromatin we've talked about in class before? There's 2 prefixes for it. What do we call DNA that's loosely packaged in proteins? That's me-chromatin. No, euchromatin, you're right. Euchromatin is DNA that's not so tightly packaged up into these proteins. So the DNA is pretty accessible to other proteins. So for example, this might be what euchromatin would look like down in the bottom right, where the DNA is associated, yes, it's wound around histone proteins, but in between the histones, there are regions of the DNA that aren't bound up with proteins. Right? The DNA is just hanging out loose in the nucleus. What's the other type of chromatin? Heterochromatin. That, we've talked about before, is what chromosomes look like when they're packaged into things in the upper left here that we know look like chromosomes when we view them at metaphase. Those are heterochromatin chromosomes, where the DNA is not only wound around histones, but the histones are packed together like they are shown here. So the histones assemble with other histones, and then those form coils, and the coils form coils, as are shown here. So the DNA is getting really, really, really densely packaged. Why does that affect gene expression? Why does being in heterochromatin, DNA packed with lots of proteins, why does that affect whether or not genes can be transcribed? What's the actual physical basis for why DNA packaging affects whether or not genes can be transcribed?
RNA polymerase can't reach the
Oh.
promoter region.
Okay, so yeah, how is RNA polymerase, if you've got an RNA polymerase molecule or protein here, yeah, how is it actually going to physically squeeze in between all of these proteins that the DNA is packed around and then actually grab onto the DNA and read it? How are transcription factors, proteins that bind to DNA, a certain sequence of DNA, how are they going to access, let's see, you've got a transcription factor, just trying to find a specific DNA sequence to bind to. When the DNA is really tightly wrapped around these histone proteins, that transcription factor has no place to grab onto the DNA. The DNA is already packaged. It's already in these huge proteinaceous blobs. So when is it, the most important thing to remember, when is it that DNA can be transcribed? Is it when it's in euchromatin or heterochromatin? Eu. Yeah, euchromatin, loosely packed DNA, means that you can have things like RNA polymerase or a transcription factor access and read DNA sequences. It's when the DNA is loosely packaged, euchromatin. The other important point to make here for understanding how epigenetics works is the structure of the histone proteins. Notice the only important point down here is the reason I put this whole slide in the first place. Histone tail. So most of the amino acid sequence in a histone is in these blobs. So the histone the linear sequence of amino acids in the histone protein folds up on itself to create a globular protein. So you could have something like histone H2A. But what happens is its N-terminal end, there's the amino end, the first amino acid, which is going to be what? What amino acid is always at the N-terminus of a protein? Start codon. What's the start codon? Methionine. Okay, so there's always a methionine at the N-terminus. And the C-terminus of the protein, the last amino acid, there isn't one. You don't know which of the 20 it is. Those two domains of the protein don't adopt any particular shape when the histone protein folds up. So we can predict what most of the amino acids in the histone proteins are going to look like. They fold up in a particular structure. But the first and the last amino acids just sort of flopping in the breeze. The tails, the N-terminal tail and the C-terminal tail of the amino acid of the protein, the amino acids just sort of hanging out. And that is where the first type of epigenetic change we're going to talk about occurs. There are proteins and enzymes in our cells that grab onto those histone tails, the N-terminal and the C-terminal tail, and they start tagging them with different chemical modifications. So we're talking about amino acid modifications. The other type of epigenetic change is not chemical modifications to amino acids, but chemical changes to the DNA itself. So there are two broad flavors of chemical changes that cause gene expression changes. One type is to amino acids, the other type is to DNA. We're going to start in detail with the DNA first. We'll go back to amino acids in a minute. Any questions, confusion, clarifications needed at this point? So we're going to talk about what chemicals are used to modify amino acids in DNA, and then we're going to immediately jump into what the pattern is. How do you know which chemical modification to which molecule turns a gene on or off? That's where we're headed. So methylation is the most common modification, chemical modification of nucleotides, A's, T's, G's, and C's. And most often, although not always, most often that happens by addition, as it says up here, of a methyl group to a cytosine, a C, that's just before a G. And I don't know really why we have to say CPG, but geneticists do. It's because that P represents the phosphate that's in the backbone between the C and the G. So there's a cytosine, right? This is the 5' end. There's a C, and then there's a phosphate group in the middle, and then there's a G, and that's the 3' end. So it's just two nucleotides. You could just call it a CG, but for some reason, say the P. It matters, apparently. So CG dinucleotides, the cytosine gets methylated. So now this C is called methylcytosine. Shocking, right? Could epigenetics get any more boring? You methylate a cytosine, it becomes methylcytosine. Go figure. How is this going to affect gene expression? What's necessary for gene expression to occur? What are some of the first steps in transcription? Tell me some proteins that are involved in transcription. Besides RNA polymerase, that's cheating. That's too easy. What? Okay, so GAL4. So we've got proteins. What does GAL4 do? Great idea. Great suggestion, GAL4. I didn't pay her to say that either. What does GAL4 do? It's got two domains, right? It's a protein that has two different parts of the protein that do two different things. What are those things? Which organism was GAL4 in? Is that in bacteria or eukaryotes? Eukaryotes. We talked about it in yeast, I think. Okay, so what does GAL4 do? This is a problem with working with tablets, right? Everyone's like flipping back through their notes. Which lecture was that? So it's important in starting transcription. Does GAL4 bind proteins or DNA? Just trying to jog your memories because this will definitely be on the final. So it binds.
It helps recruit RNA.
It recruits RNA polymerase.
Yeah, look at it.
Yes. I knew that was what you meant. That's why I said it. Good job. So yeah, Gal4 is important in bringing RNA polymerase to the DNA. So what does Gal4 have to be able to do to bring in or recruit a polymerase? How does Gal4 know where to put the polymerase? UAS. So Gal4 has a protein binding domain, right? 3 minutes ago, which seems like forever ago, I asked what are the two roles of Gal4? What two functions does it have? One is it binds proteins, like it brings in RNA polymerase. What other kind of molecule does Gal4 bind to regarding the UAS? Well, what is a UAS? Upstream activation what? Sequence. What kind of molecule does Gal4 bind? DNA. Right. So Gal4 is a transcriptional activator. It's a protein that binds to DNA. It grabs onto RNA polymerase and says, here, this is where you start transcription. So Gal4 recognizes a specific DNA sequence. That's the UAS. How could methylation of DNA affect gene activation? How could that disrupt that process of Gal4 binding its DNA sequence, grabbing onto RNA polymerase? Because the sequence changes with the methylation. Well, it's still a cytosine, but it does something. Is everybody familiar with the word steric? Stericking at you right now. No. What does steric mean? Steric hindrance. Right? Oh man, everybody's having flashbacks. Right? Crowding. Physical, keeping something away physically. I could use my tablet to sterically hinder you from throwing tomatoes at me at this point. Methylation prevents, for example, transcription factors, DNA binding proteins, from binding to the DNA. It's causing a physical change in the shape of the DNA. The DNA has all these little chemical bits sticking off of it that the proteins that recognize that sequence don't know what to do with. Right? And it's a minor difference. Right? This is the difference between cytosine and methylcytosine. Not shockingly, I know. There's a single methyl group, as we'd already talked about. But that doesn't make much of a structural change. It's a tiny little change, but it's of great importance because that sort of change does prevent proteins from binding to DNA sequences. And that's how expression of genes is affected. So here's a cartoon example. You get methylation of one CPG, one CG dinucleotide down here, and that prevents a transcription factor from recognizing and binding to that sequence. So transcription doesn't occur. Effect, methylation, I'm slowing down and saying this because I want you to remember this part. Methylation leads to inactivation. Right? When you have those methylations of CPGs, the cytosines specifically, in that two nucleotide sequence, CG. C gets methylated, prevents things from binding to the DNA, prevents transcription. Gene inactivation. Not changes to the DNA sequence itself, modifications to existing nucleotides. So you add a little tiny methyl group, you turn off transcription. Now histones, like DNA, can have some of the same types of modifications. Remember I talked about the histone tails, the N-terminus and the C-terminus. We've got 4 different types of histones, H2A, H2B, H3, and H4. Each of them have an N-terminus and a C-terminus. So there are 8 different tails in a nucleosome that could be chemically modified. They get both methylated like DNA does. So methyltransferase enzymes can actually also add methyl groups to amino acids that are part of the histones. They also get acetylated. So you can have methylation, the addition of a methyl group, or you can have the addition of AC, an acetyl group. And those can happen anywhere on histones, although most usually those happen on a specific type of amino acid, one of the 20, lysine. Not always, but very often these modifications happen on amino acid K, lysine. So lysine happens to be the favorite amino acid of the enzymes that stick these chemical groups on. I guess I should have put this slide earlier. There you go. 8 histones, 2 tails per histone. So there are a lot of targets for these enzymes to come add chemical modifications. Now, how would you predict chemical modifications of histone tails would affect transcription? So when modification occurs to the DNA itself, that prevents proteins from binding to it. When modifications occur to histone tails, what does that do? We'll get there in a second. Please don't panic. You already know that you don't have to memorize a lot of stuff because you've got access to this when you need it, but here are diagrams of the amino acid sequences in humans anyway, of the histone tails. Right, H2A, H2B, H3, and H4. Look how many lysines there are. It's almost unfair. So many Ks in these tails. And these enzymes can add methyl groups into acetyl groups all over the place. And remember that these amino acids are numbered starting from the N-terminus. So if you blow this up, you can see that serine, in this case, not methionine, is the first amino acid in the N-terminal tail of this protein. Right? So this lysine, what's its number? Yeah, this is lysine 8, there's lysine 5, there's lysine 12, right? A lot of lysines. The histone code that's on this table refers to these lysines both by which histone it is, H2A, H2B, H3, and H4, and then which lysine in which tail. Is it the N-terminus or the C-terminus? And is it lysine 5, 6, 7, 8, whatever? So that's how you read these headers right here. So this is histone H3. It's the lysine that's at the 4th position, H3K4. So we start at the 1st, 2nd, 3rd. It's the 4th amino acid in on the N-terminal tail of histone H3. And then the rest of this table tells you whether or not different modifications, methylations or acetylations, are known to generally, not specifically, but generally, are they known to activate or repress transcription? So I ask you, what's the pattern? Take 10 or 15 seconds, look at the table. What sort of chemical modifications tend to activate or repress? Is there a pattern? So if you had to say one of these types of modifications of the 4s shown, which one is most likely to repress transcription? Yeah, trimethylation, I heard. Right. Quite a bit of the time, trimethylation at certain histones, like H3K4, H3K9, if you not H3K4, that's activation, H3K9, lysine 27, lysine 79, generally speaking, if you found a modification of 3 methyl groups on that lysine, that would lead to, or is correlated with, repression. Acetylation leads to activation. And acetylation only happens on certain lysines, on certain histones, but when you find it, that's typically linked to that gene as being actively transcribed. This is referred to generally as the histone code. People are getting Nobel Prize for it, and this was discovered about 15 years ago. Nobody had really studied chemical modifications to the proteins that DNA is packaged with, but those modifications, the epigenetic marks, the additions of methyl groups and acetyl groups and others we're not talking about yet, are critical for gene expression regulation.
So do the blank spots means that no change occurred?
Right, so the blanks in the table
Yeah.
means either there's no clear pattern or that type of modification doesn't occur. Right. So for example, as you were probably thinking of, histone 3 lysine 14 never gets methylated, it only gets acetylated. And if it's unacetylated, if nothing has happened to it, if it's just lysine, then you can't predict what the gene's doing. But when that lysine's acetylated, that usually means, 99% of the time maybe, that the gene that is wound up in the histones in that part of the chromosome are active. So you can imagine this is a really dynamic thing, or I hope you will imagine. Imagine with me. Close your eyes. Don't fall asleep. You've got a huge chromosome. Loads of nucleosomes. Millions of nucleosomes per chromosome. So there are certain regions, this is what I'm hoping you're imagining, there are certain regions of chromosomes where histones are acetylated. There are other regions of the same chromosome where the histones might instead be methylated. Parts of chromosomes where the DNA is methylated at CPGs. And then way down there, the CPGs aren't. So these are not modifications that happen every location in the genome all at once. Because if everything was being methylated at the same, if all the DNA was being methylated at the same time, that would turn off all transcription, right? What gene expression regulation is all about is finely tuning gene expression. So you turn off some genes in certain cell types, but you want to turn on others. So this is not just regulation cell to cell, this is regulation of genes that occurs on a chromosomal region by chromosomal region basis. Which is easy to imagine, and then it's hard to reconcile with how does that actually work? You've got enzymes in the cell that are going around, and every time they see a CPG, they add a methyl group. And they do this all over the place. So what has to happen inside the cell to prevent everything from being methylated all at the same time? I'm a methylase. How are you going to stop me? What's the cell going to do? How is the cell going to make sure that the methylase only methylates the parts of the nuclear genome that it's supposed to? Think superhero movies. Think current superhero movies. Okay, imagine that the methylase is Iron Man. What other sort of enzyme is going to be in the cell? Superman, right? No, I don't know. Batman, Superman, Iron Man. Who cares? What other enzyme is in the cell? Besides the one that's putting on methylation on CPGs. What's the opposite of that? You've got methylases. So you've got enzymes that add methyl groups, and then you've got enzymes that are taking them off. So I'm over here working, trying to add methyl groups. But the way that this is regulated is that the cell also produces methylases that remove methyl groups from CPGs. So the imaginary scene that we're playing out in our minds is getting even more complicated. Chromosomes are actively methylating certain parts of their DNA and actively demethylating, removing methyl groups from others. And the same is true for acetylation and methylation on histones. Methylation of histones tends to lead to compaction of the histones. That's mediated by other proteins that recognize those methyl marks and sort of seal them together or bind them together. So methylated histone tails, structural proteins grab onto and keep those histones close to each other. Compaction, heterochromatin, genes turned off. When those methyl marks are gone and when acetyl groups are present, those proteins don't bind to unmethylated or acetylated histone tails. Those structural proteins that would cause condensation of the chromosome leave the histones and the nucleosomes alone. So the DNA is a little bit more open, its gene's expression goes up. So methylation, if there's a trend, and I'm not saying there is one, but if there is one, if you go back to that histone code table, it's unclear that there's a trend. But if there is one, it's that methylation, either DNA or histone, reduces transcription. Methylation, gene expression goes down. Acetylation doesn't happen to DNA, but if acetylation happens on histones, that's usually a mark of activation. Methylation, decreased transcription. Acetylation, histones only, increased transcription. Any questions? With that in mind, we're going to move on to an exercise for you to try to apply this information. It's an easy one. Should go pretty quickly. So now with the background in epigenetics, just a little bit of information. How it works, we saw the movie at the start of class, random X chromosome inactivation, we're about to come back to. That's the case study we'll talk about today. The next couple of classes will talk about whether or not epigenetics affects sexual preference. And then we'll also talk about relationship of epigenetics to asthma. So X chromosome inactivation. Started with a cat, we end with a cat. For you. All right. Calico cat. The start of the movie, or start of class, we saw a movie that said that X chromosomes get randomly turned off in females at a certain age during development. So if you're a if you have two X chromosomes and one of them, there's a subtle distinction here, have the capital OR allele of the coat color gene, that produces orange pigment. And if you have the little o version, those cells will produce black pigment. So this is how tortoiseshell or calico cats occur. One of these two X chromosomes gets turned off. So if you're a heterozygote, what's going to happen to your coat color? Let's say this is a diagram of a cell that came from a black part of the cat. So this is a black pigment producing cell. Which of these two X chromosomes would have been inactivated in that part of that female cat? X1 or X2, if it's producing black pigment? Right. X1 became Xi, the inactive X. Okay. That means that this chromosome is a blob. The capital O, orange-producing allele, doesn't get transcribed. Proteins can't access it, transcription factors, RNA polymerase can't get in there and actually transcribe the gene that makes the protein that produces orange pigment. What sort of methylation and acetylation marks will be on the Xi? X1. What will we expect? What do you predict? If it's the one that's being turned off. So I heard trimethylation. Me. It's all about me. Which sort of molecule is trimethylation occurring on? Of DNA or amino acid? Trimethylation, 3 methyl groups on the same spot, only happens in one of the two. It's either DNA or it's in lysine. Flip back to the table if you're not sure. It's lysine. So histone modifications, you can get 1 methyl group, 2 methyl groups, 3 methyl groups. So you'd expect a lot of lysine trimethylation on the X chromosome that becomes inactivated. That's after it's inactivated, by the way. So the trimethylation is the signal to turn that chromosome off, to turn it into heterochromatin, to make it a bar body. What modification to DNA would you expect on this chromosome? So we've got some C's and G's. What's going to happen? The inactive X. What mark goes there? What's the only chemical modification that epigenetic chemical modification that occurs to DNA? Is it methylation or acetylation? It's methylation. So you methylate C's in CPGs, so you get methylcytosine. That's also a signal that a chromosome is, or a gene or a region of a chromosome has been inactivated. Okay. What would you predict on the other chromosome, the one that stayed active? What sort of modifications does it have? We better have the other type, or else I wasted a lot of time talking about it. What happens on the active X chromosome? What chemical modification? Yeah. Say it loud and say it proud. Lysine acetylation. And the opposite pattern would be true if you want to do this for practice. What does a cell that's producing orange pigment look like? Which X chromosome gets turned off and which chemical modifications will occur on those two? You can do that on your own, but I'll tell you now it's the opposite pattern. So all of these modifications that you find on inactive X chromosomes are associated with silence. So every female body has parts, patches, like a mosaic, that have one X chromosome turned off or the other. Like the calico cat, like this actual mosaic of a female bust. What dictates how big these patches are? That is how mosaic a female body is. Whether or not it's little patches on the body that have X1 or X2 turned off, or big blocks of tissue. What affects the pattern? We saw this at the start of class in the video. How did this, now we're eating into Socratic time. Right, here's a patch of tissue, presumably, that all arose from what? All of these cells, how are they related to each other? They all have the same X chromosome inactivated. What does that mean about those cells?
They all came from the same cell, like early embryonic development.
Right, they all came from the same initial cell that turned off one or the other of its X chromosomes. And then during cell division and cell division and cell division and cell division, you produce one part of an adult body. So that whole group of cells, all of which have the same X chromosome turned off, all originated from the one cell where the X chromosome inactivation actually occurred. Which one randomly got turned off? So what does it tell you if you looked at this, if this was an actual individual in, say, a calico cat? What does it tell you how big these patches are, or how many patches are on the body? What does that tell you about the X chromosome inactivation process? How many cells would you estimate in this actual mosaic on the right? How many cells would you suggest were in the embryo when X chromosome inactivation occurred? How many patches are there? Like, I don't know, what do you say?
Like 15.
15 or 20? Okay. So that suggests, in this case, that it was maybe the 16-cell stage when X chromosome inactivation occurred. So when the embryo there was fertilization, one division produces 2 cells, the second division produces 4, then 8, then 16. So that would tell you that it was about that stage during development when each independent cell in the embryo chose which of its X chromosomes to turn off. If X chromosome inactivation occurs later during development, how would that affect this pattern? What would the torso look like instead? If instead of the 16-cell stage it was the 256-cell stage, that those cells randomly chose an X chromosome to inactivate, how would you draw what that would look like? How would this change? Yeah, you'd have smaller patches. There would be smaller spots in the body, each of which are derived from that individual one of 256 cells that were present in the embryo when X chromosome inactivation occurred. So next time when we get together, still a Socratic quiz. I'll leave it open for the afternoon so you've got time to finish it. It only asks a couple of questions. They're easy. Next time we get back together, we're going to find out whether or not epigenetics, chemical modifications that occur after birth sometimes, can affect sexual preference in humans. So head on to Socratic quick quiz whenever you like this afternoon. I'll turn it off maybe 3 o'clock. I'll see you next time.